Good day, this is Professor Will, CBMD PhD. Today is Sunday, June 23rd, 2019. It is 2:23:15 seconds Eastern Standard Time, and this is a continuation of my scientific research, so let's get started here. Sugars that coat proteins are a possible drug target for pancreatitis. Date June 20th, 2019. Source Cold Spring Harbor Laboratory. Summary: CA19-9 is a complex sugar structure that coats proteins. Elevated levels of CA19-9 was found to cause inflammation in the pancreas in mice and promote rapid progression to pancreatic cancer. By neutralizing the functions of CA19-9 with antibodies, researchers were able to reduce and even prevent pancreatic damage in animal models. Researchers have focused on a potentially powerful biomarker, a chemical structure created by a complex sugar molecule called CA19-9. Since CA19-9 is elevated in patients with pancreatitis and pancreatic cancer, the research team provided the first evidence that the CA19-9 actually causes the disease it was correlated with as a biomarker, and suggests that blocking this complex sugar structure could be used therapeutically to prevent the progression from pancreatitis to pancreatic cancer. This is one of those unique opportunities where prophylactic intervention of pancreatitis may lead to prevention of pancreatic cancer in at-risk patients. Researchers zeroed in on CA19-9, a complex sugar structure that coats many proteins but had not previously been ascribed with any particular function. A single enzyme controls the final step in production of CA19-9 in humans, but this enzyme is missing in rodents. Researchers generated mice that produce CA19-9 and surprisingly noted that the mice developed severe pancreatitis. Research findings position CA19-9 as an attractive therapeutic target for pancreatitis. In mice, CA19-9 recruits the immune system to repair injuries from pancreatitis. Researchers found that during this recruitment process, CA19-9 can also induce a cascade of biochemical reactions propelled by the release of deleterious digestive enzymes from the pancreas. This cascade opens a transformational gateway for cancer to develop. Researchers also demonstrated that CA19-9 can dramatically accelerate the growth of pancreatic tumors. Pancreatitis is required for developing pancreatic cancer, and we might be able to prevent that transition in patients with pancreatitis by targeting CA19-9. By targeting CA19-9 with antibodies in animal models, we were able to reduce the severity in pancreatitis and even prevent it from occurring. Today, this is Professor Will, CBMD PhD. Today, Sunday, June 23rd, 2019. It is 2:52:40 seconds Eastern Standard Time. This is a continuation of my scientific research, so let's hop to it here. Treatment for common cause of diarrhea, more promising intestinal parasite Cryptosporidium successfully grown in lab using new technique. Date June 20, 2019. Source: Washington University School of Medicine. Summary: researchers have figured out how to grow the intestinal parasite Cryptosporidium in the lab, an achievement that will speed efforts to treat or prevent diarrhea caused by the parasite. New researchers at Washington University School of Medicine in St. Louis have figured out how to grow the most common type of Cryptosporidium in the lab, a technological advance that will accelerate efforts to treat the deadly infection. This parasite was described over 100 years ago, and scientists have never been able to reliably grow it in the lab, which has hampered our ability to understand the parasite and develop treatments for it. We now have a way to cultivate it, propagate it, modify its genes, and start figuring out how it causes disease in children. This is a first step towards screening potential drugs and finding new drugs or vaccine targets. In wealthy countries, Cryptosporidium is notorious for causing waterborne outbreaks of diarrhea. The parasite goes through a complex life cycle, including a stage in which it is called an oocyst and becomes hardy, spore-like, and hard to kill with chlorine, bleach, or other routine sanitation measures. In 1993, 400,000 people in the Milwaukee area developed diarrhea, stomach cramps, and fever after a malfunctioning water purification plant allowed Cryptosporidium into the city's water supply. Every year, dozens of smaller outbreaks are reported in the U.S., many associated with swimming pools and water playgrounds. Diarrhea caused by the parasite can last for weeks. While this is miserable for otherwise healthy people, it can be life-threatening for undernourished children and people with compromised immune systems. Until now, researchers who wanted to study the parasite had to obtain oocysts from infected calves. Cryptosporidium infection is a serious problem in commercial cattle farming, and grow the parasites in human or mouse cell lines. The parasite inevitably would die after a few days without going through a complete life cycle, so the researchers would have to obtain more oocysts from cattle to do more experiments. Researchers suspected that the problem lay in the cell lines traditionally used to grow the parasite. Derived from cancer cells, these cell lines were very different from the normal, healthy intestine that is the parasite's usual home. Researchers and colleagues cultured intestinal stem cells to become mini-guts in a dish, complete with all of the cell types and structural complexity of a real intestine. When the researchers added oocysts to the mini-guts, the parasites thrived. They emerged from the oocyst and went through their full life cycle to produce more oocysts. For the first time, every stage of the parasite's complicated life cycle could be studied in the lab. The researchers also showed that they could edit the parasite's genes with CRISPR-Cas9 and perform genetic crosses, making these powerful tools for studying biology more accessible than before. We put the parasite in this environment that is much more natural, and it is happy, and it grows and develops and goes through the entire life cycle. This opens up possibilities that were closed for a long time. There is only one FDA-approved drug, and it doesn't work in young children. There are potential drug candidates, but we couldn't screen them before because the parasites would just die away. How can you tell if the drug is killing the parasites if they are already dying? Now we can start screening drugs and also start asking questions about what makes this parasite dangerous. The technique applies only to C. parvum, one of the most common cryptosporidium species that cause diarrhea in people. Its cousin C. hominis is even more difficult to grow in the lab. The two are closely related, but while C. parvum can infect young mammals of many species, C. hominis only infects people. There are only a few dozen genes that are different between parvum and hominis, but somehow that is enough to make hominis very finicky. It won't grow in mice or in calves, or so far in mini-guts grown from mouse stem cells. Developing systems to work with hominis is an important goal in the lab. The technique, while potentially powerful, will not immediately translate into better treatment or prevention for diarrhea. These things take time. There's a lot of basic research that still needs to be done, but this system provides an important path forward. We can now use genetic approaches to study the role of individual genes and thereby identify important targets for improved therapies. Good day, this is Professor Will, CBMD PhD. Today is Sunday, June 23rd, 2019. It is 3:29, 3 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's hop to it here. Scientists discover new methods for developing tracers used for medical imaging. Date June 20, 2019. Source UNC Limburger Comprehensive Cancer Center. Summary: researchers discovered a method for creating radioactive tracers to better track pharmaceuticals in the body as well as image diseases, such as cancer and other medical conditions. The research reported in the Journal of Science a method for creating tracers used with positron emission tomography or PET imaging. Researchers said their findings could make it possible to attach radioactive tags to compounds that previously have been difficult or even impossible to label. Positron emission tomography is a powerful and rapidly developing technology that plays key roles in medical imaging as well as in drug discovery and development. This discovery opens a new window for generating novel PET agents from existing drugs. PET scans track a radioactive tag that is attached to a compound. These tracers are generally injected into the body, and they produce bright images on medical scans as the tracer accumulates in the target lesion, organ, or tissue. Scientists can attach tags to molecules like glucose, which will accumulate in tumors as cancer cells consume sugar to drive their overreactive growth, or to amino acids, which, as the building blocks of proteins, can be highly consumed in tumors. They can also attach them to potential new drugs to track their course in the body. What is believed to occur is that tumor molecules uptake these resources faster than healthy cells do. What we've contributed to the field is a new method to introduce radio-labeled isotopes of atoms into drug molecules in a way that hasn't been done before. In their study, the researchers described a new way of attaching a radioactive molecule, fluorine-18, a widely used isotope in PET imaging, by breaking a specific chemical structure of carbon and hydrogen atoms. In the presence of blue light from a laser and after the addition of catalyst material to speed the reaction, the researchers could break existing chemical bonds in the structure and insert fluorine-18. Once attached, the tracer emits gamma rays that are picked up by imaging. The researchers used a cyclotron, a particle accelerator, in UNC's Biomedical Research Imaging Center to create fluorine-18. Researchers envision multiple potential applications for their discovery, including for medical imaging to screen patients for a response to a drug, or to aid in drug development research. Not only can we study where drugs are localized in the body, which is something that's important for drug and development work, but we could also develop imaging agents to track cancer progression or inflammation in the body, aiding in cancer research and Alzheimer's research. Having more than one method for tumor detection may give you cross-verification to make sure that you are seeing is real. If you have two methods to validate a scan, two is better than one. While existing radio labeling methods requires the synthesis of dedicated new compounds to attach the radio tag, researchers say their approach may allow them to attach the tagged existing compounds a boon for drug development research. In this study, we showed that we could label a broad spectrum of compounds, or anti-inflammatory drugs, and specific amino acids to show that they could image tumors. Researchers also said the information obtained by the new PET tracer could aid in development of corresponding treatment plans depending on the image results, which would be an important step in providing personalized medicine. The researchers said the next step is to develop a device that would make it easier for scientists to use this new method for creating radio-labeled tracers. In addition, they are working to expand their technology to develop other tracers that use a different radioactive material such as carbon-11. This discovery opens a new window for generating novel PET agents from existing drugs. Many very complicated or almost impossible to label drugs could potentially work using this method. Good day, this is Professor Will, CBMD PhD. Today is Sunday, June 23, 2019. It is 3:53:28 seconds Eastern Standard Time. And this concludes my scientific reading. Hope you enjoyed listening. Have a great day.